Hello friends, good afternoon and welcome to EduSat Live Lectures. Dear friends, today in genetics we will be talking about explaining genetics using probability. To discuss this topic we have with us our subject expert Dr. Varunendra Singh Rawat. Dr. Rawat is Assistant Professor in Department of Zoology, Hindu College, University of Delhi. Without further ado, I would like to welcome Sir to our studios and request him to start the lecture. Welcome,
Thank you.
sir.
So friends, today I'll be talking about probability and its uses in genetic events. Probable is a term which we use very commonly in our day-to-day life. For example, if we say that it might probably rain today. So what does probable mean? Probable means it talks about certain event which is likely to occur. But when we are talking about probability in general life, we do not add any mathematical connotation to it. But in genetics, we use mathematical formulas to give a specific value to any event, likelihood of any event which might occur. So today's lecture will be dealing with probability and how it can be used in explaining genetic events. The outline for today's lecture will be I'll be talking about meaning of the probability. What are the different rules which govern the calculation of probability for different events? I'll be talking about conditional probability and binomial theorem and its use in calculating probability of the genetic event. Then we'll be solving some problems which are based on probability. So the learning objective for this particular lecture would be to calculate the probability of occurrence of a genetic event using the rules and formulas of probability. Now what exactly probability means in mathematical terms? Probability is the study of the likelihood of occurrence of a particular event. The events which are certain to occur have a probability of occurrence 1. That means the events which are going to occur, they will be given a probability score of 1. The events which can never occur have a probability score 0. All the other events which fall in between will be having probability value between 0 to 1. So all the events which have a variable chance of occurrence have the probability value lying between 0 and 1. For example, if we toss a coin, the probability of getting heads on tossing a coin is 1 by 2 because we can either get head or we can either get tail. So for getting head, the probability is 1 by 2 which equals to 0.5. That means every time we toss a coin, the chances of getting head is 0.5 and the chances of getting tail is 0.5. Some biological processes seem to be directly affected by the chance outcome. And these processes are namely, example, formation of gametes, recombination events, etc. So a biologist must have the knowledge of calculating probability for these events. And in today's lecture, we'll be dealing about getting the basic idea of probability calculation. The probability can be either empirical or theoretical. Empirical probability of an event can be calculated by dividing the number of times an event occurs by the total number of times that event could have occurred. For example, in Mendel's experiment, if green colored seeds occurred 1424 times out of 8000 times, then the probability of occurrence of green seeds is 1424 divided by 8000 which equals to 0.178. So the probability of occurrence of green seeds in Mendel's experiment is 0.178. So basically empirical probability is dividing the number of times the event has occurred and it is dividing it by the number of times that event could have occurred. Whereas theoretical probability of event can be calculated by dividing the number of times an event is expected to occur by the total number of times that event could possibly occur. For calculation of theoretical probability, the knowledge of rules which are governing that particular event is essential. So we require to know the knowledge we require to know the rules which are governing that particular event in order to calculate the theoretical probability. For example, a mating between parents which are heterozygous for albinism alleles. They have one capital A allele and one small a allele. Both the parent, that is father and mother, have capital A and small a in their genotype. When they mate, it leads to individuals or offsprings which have genotypes capital A, small a and small a, small a. The probability of recessive homozygous individuals being born in this particular event can be found by using Punnett square. As seen in this particular slide, the gametes which are formed from father and from mother, they might be having either capital A allele or small a allele. When these gametes combine randomly, they give rise to 3 types of genotype, and the frequency of occurrence of each genotype varies. Now, for this particular mating between heterozygous parents and for producing individuals with which are albino, that means which are having genotype small a, it can occur only if a sperm carrying small a allele fertilizes an ovum carrying small a allele. So, the probability of formation of gamete with capital A or small a is 1/2. Because the parent is heterozygous, the gamete formed will be either capital A or it will be small a. So, the probability of formation of gamete with small a is 1/2 in both the parents. Therefore, probability of formation of a recessive homozygous individual with the genotype small a can be calculated by multiplying the 2 individual probabilities, which is 1/2 into 1/2, that is 1/4. So, out of 4 offspring which are born to this particular couple, the chances of having an albino child is 1, that is 1 out of 4 child might be albino, and it equals to a probability of 0.25. Larger the data used to calculate the empirical probability, more closely will it approach the theoretical probability. So, in the previously mentioned example, if only 2 offsprings are born, then achieving the probability of 0.25 for an albino child might not be met with. But if the number of children are more, suppose 8 children are born, then we can see that the probability of occurrence of albino child, that is 0.25, will be met in this particular example. So, we need to have a large data for matching the probability of any event. The certain rules are there which govern the calculation of probability in any of the genetic event or any of the mathematical event. Herein we are using the probability for calculating the likelihood of any event in genetics. So, there are 2 particular rules, namely product rule and sum rule. The product rule states that the probability of occurrence of 2 independent events together can be calculated by multiplying individual probabilities of the event. Events are said to be independent when the outcome of each event does not affect the outcome of other events which are also under consideration. So, the essential condition for applying product rule is that the outcome of 2 events is not dependent on each other, they are independent of each other. For example, if we toss 2 coins, we have one 1 rupee coin and a 5 rupee coin, and these are tossed together. There can be 4 possible outcomes of this tossing. The two coins are getting tossed together simultaneously. The first possible outcome is that the 1 rupee coin will turn up with tail and the 5 rupee coin will also turn up with tail. The probability of getting a tail is 1/2 in both the coins. So, the total probability for this event will be 1/2 into 1/2, that will be 1/4. So, the chance of getting 2 tails when we toss 2 coins is 1 out of 4. The second possible outcome is the 1 rupee coin will get tail and the 5 rupee coin will get head. Again, since for getting head and for getting tail, the probability is 1 by 2 for both the events. So, for this particular event, the probability will again be 1 by 2 into 1 by 2, which will be 1 by 4. The third possible outcome is that both the 1 rupee coin and the 5 rupee coin, they turn up with head. And again in this case, the probability will be 1 by 2 into 1 by 2, which again is 1 by 4. And lastly, the 5 rupee coin might turn up with tail and 1 rupee coin might turn up with head. And again, the chances of occurrence of this particular event is 1 by 4. So, all these 4 events can occur 1 out of 4 times. So, in this particular example, the probability of getting head or tail is 1 by 2 and all the 4 possible combinations are possible, and the probability of all the 4 possible combinations is 1 by 4 each. Now, if we consider this particular product rule in using a genetic example, now for suppose if a mating occurs between individuals which are heterozygous for cystic fibrosis gene, then what is the probability of an afflicted child being born? Now, both the parents, that is male and female, they are heterozygous. The genotype is capital C and small c for both, and both will contribute small c allele to 1/2 of their gametes because they are heterozygous. So, the probability of afflicted individual being born in this particular case will be product of the individual probability of the gamete being carrying the small c allele. So, the ovum might carry small c allele 1 out of 2 times and the sperm might carry small c allele 1 out of 2 times. So, the final probability will be 1 by 2 into 1 by 2, that is 1 by 4. So, for a heterozygous couple for cystic fibrosis gene, the chances of producing a child which is homozygous recessive having genotype small c will be 1 out of 4, that is 0.25 times. Now, other rule which governs the calculation of probability in genetic events is sum rule. For calculating the probability of a generalized outcome that can occur in more than one way, sum rule is used. In the earlier example, that is of the tossing of 2 coins together, what is the probability of tossing a 1 rupee coin and a 5 rupee coin and getting 1 head and 1 tail? Here we are not specifying which coin should get head and which coin should get tail. We are just stating that we want 1 head and 1 tail irrespective of the coin. So, what are the chances for this particular event to occur? Now, the head and tail combination might occur if 1 rupee coin is having tails and the 5 rupee coin is having head, or it might also occur if the 5 rupee coin is having tails and 1 rupee coin is having head. Both these combinations are possible. In each case we are getting 1 head and 1 tail, and for each of the event the probability, individual probability is 1 by 2 into 1 by 2, that is 1 by 4. So, both the events are possible with probability 1 by 4, but we want to find out how many times we will get 1 head and 1 tail irrespective of the coin which is showing head or which is showing tail. In that particular case we will be using sum rule. So, we will be adding up the 2 individual probabilities of these 2 events, which is 1 by 4 will be added to 1 by 4 and we will be getting probability of 1 by 2. That is, whenever we toss 2 coins, the probability of having 1 head and 1 tail is 1 out of 2, that is 0.5 times this is going to happen. We will be getting 1 head and 1 tail. Now, using the sum rule in the example, in the genetic example, if we consider a mating between two individuals which are heterozygous for cystic fibrosis gene, that is they have capital C and small c genotype, then what is the frequency of occurrence of a heterozygous child? that is a child which will be having genotype capital C and small c. Now, two events can lead to a heterozygous individual. These two events are possible when a gamete carrying capital C allele from male and a gamete carrying small c allele from female fuse together to give rise to an embryo. The probability of a sperm carrying capital C allele is 1 by 2, and for carrying small c allele is also 1 by 2. So, the total probability is 1 by 4, whereas gamete C from male and small c from male and gamete capital C from female will be also be 1 by 4. So, overall probability for this event of birth of a heterozygous child will be 1 by 4 plus 1 by 4. So, it will be 1 by 2. So, these are the two rules for probability, and these can be used for calculating probability in genetic events. So, there are two rules, namely product rule and sum rule, which can be used to calculate probability of any genetic event, like calculating genotype of a offspring being born in a family. Probability is useful in predicting the chances of occurrence of an offspring of a given genotype in family. However, this prediction of possible outcome is only realized when the sample size is quite large. In a small sample size, it might not be giving an accurate probability. Another way to calculate probability of events is by using conditional probability. Conditional probability is used to calculate the probability of an outcome which is dependent on a specific condition related to that particular outcome. For example, in Mendel's monohybrid cross, what is the probability that F2 tall individuals which are born after the cross of heterozygous individual, what is the probability that F2 tall individuals are heterozygous and not homozygous? So, we are setting a condition to this particular event, and the condition which is set is only tall plants are being considered. Both the probability of the outcome of interest and that of specific condition which include the outcome should be calculated for finding out conditional probability in this particular event. The probability of occurrence of heterozygous individuals is 1 by 2 because both the parents are heterozygous. Secondly, probability of occurrence of tall plants is 3 by 4, as we saw in the Punnett square. 3 out of 4 individuals will be born tall. The conditional probability for this particular event, that is the tall plants are heterozygous, can be calculated by dividing the probability of occurrence of heterozygous individuals from probability of occurrence of tall plants, which is 1 by 2 divided by 3 by 4, which leads to probability of 2 by 3. So, the conditional probability has a very good application in the field of genetic counseling, and it can be used by medical practitioners. Now, binomial theorem can also be used to calculate probability of events where one of the two alternative outcomes is possible during each of a number of trials. Using the binomial theorem, the probability of any specific set of outcomes among a large number of potential events can be easily calculated. For example, in a family of a large size, the probability of any combination of male and female children can be easily calculated using the binomial theorem. The expression of binomial theorem is a plus b raised to power n, which is equal to 1, where a and b are respective probabilities of the two alternative outcomes for that particular event, and n equals the number of trials which are being carried out. For each value of n, the binomial must be expanded using the following formula: a plus b raised to power n, which equals to a n plus a raised to power n minus 1 b plus a raised to power n minus 2 b square, so on and so forth till the value of b raised to power n is reached. The numerical coefficient preceding each expression can then be easily determined using Pascal's triangle. The Pascal triangle is basically as given in this particular figure. For a given value of n, the number of there will be a specific number of coefficient, and the value of exponent can be found out using this particular Pascal triangle. For example, if we have n is equal to 2, then the value of coefficient will be 1, 2 and 1. So a plus b square will be a square which has value 1 plus ab which has value 2 and b square which has value 1. So we can use this particular triangle for n number of possible outcome. Now using the binomial theorem, we can find the probability of having 2 boys and 3 girls in a family of 5 children as an example. Probability of a male being born in any family is 1 by 2, and probability of female being born is also 1 by 2. So A is equal to 1 by 2, B is also 1 by 2. The appropriate term in the expanded binomial a plus b raised to power 5 can be found out by using the formula, and as seen in this particular slide, a plus b raised to power 5 has a term 10 into a square into b cube, where a is the boy and b is the girl. So 10 a square b cube will give us the probability of 2 boys and 3 girls being born in a family of 5 children. When we put appropriate values here, we get 10 into 1 by 2 raised to power 2 into 1 by 2 raised to power 3, which comes to a probability of 5 by 16. So out of all the families with 5 children, 5 out of 16 are predicted to have 2 boys and 3 girls. This is the probability of having 2 boys and 3 girls in a family of 5 children. Now a single formula can also be applied to determine the probability of specific set of outcomes among a large set of possible outcomes, and it is basically p is equal to n factorial divided by s factorial into t factorial into a raised to power s into b raised to power t, where n is equal to total number of events, s is equal to number of time outcome A occurs, and t is equal to number of times outcome B occurs. For example, in a family of 4 children, what is the probability of having 3 daughters and 1 son? Using this formula, p is equal to n prime s divided by s prime t prime, we put the values. P is equal to 4 factorial divided by 3 factorial into 1 factorial, whole multiplied by 1 by 2 raised to power 3 into 1 by 2. 1 by 2 raised to power 3 for daughters and 1 by 2 for son. So the value comes, final value which comes is 1 by 4. So in a family of 4 children, probability of having 3 daughters and 1 son is 1 out of 4. Now what is the probability that a family of 4 children will consist of at least 2 daughters? When we talk of at least 2 daughters, we have to consider 3 possible cases: when there are 2 daughters, when there are 3 daughters, and when there are 4 daughters. We will be using this particular formula in all the cases and putting the values. For a specific case, we get 3 different probabilities. All of these probabilities will be added up for finding out the probability of having at least 2 daughters, which comes out to 11 by 16. Now what is the probability that a family of 5 being tested for PTC testing have 2 taster boys, 1 taster girl, 1 non-taster boy and 1 non-taster girl in that order? In this particular case, order is being given. We know probability of girl being born is 1 by 2, also for boy is 1 by 2, and for a taster being born is 3 by 4 in the case of PTC testing, and a non-taster being born is 1 by 4. So we will be calculating individual probability for all the events, and that will come to 1 by 2 into 3 by 4 square, again 1 by 2 into 3 by 4, 1 by 2 into 1 by 4 and 1 by 2 into 1 by 4, which will be 27 divided by 32768. This will be the probability. Now I'll be talking about pedigree analysis in genetics. It is a very important topic, and it is used regularly by geneticists and medical practitioners for advising couple who are about to have children. Now what exactly is pedigree? I'll be talking about later on. Before that, I will like to give a brief outline of the lecture, this particular lecture. So I'll be talking about meaning of probability in this particular lecture. I'll give a detailed picture of the symbols which are used in constructing a pedigree. There will be a talk about different types of inheritance which can be assessed through constructing pedigree charts, and then we will be doing some problems on pedigree analysis. So, the learning objective for this particular lecture would be to assess, we should be, after this particular lecture, we should be able to assess the type of inheritance by analyzing the given pedigree. Before I move on to the lecture, I would like to present Queen Victoria, who was the Empress of England, and she was also called the Empress of India, because during her reign only the Britishers were able to capture India. Now, apart from her reign, she is also famous for a particular disorder which she passed on to her offspring, and this disorder spread it throughout the families, throughout the royal families of Europe. And how this was found out, that Queen Victoria was the one who passed on the disorder. It was found out by constructing a pedigree chart. It was not a real time chart, but using data and using news, this particular chart was constructed, and it was seen that hemophilia was passed on in the royal family of Europe, ranging from France to Russia to England, because of a mutation in the genotype of Queen Victoria. So basically, this particular example is associated with the royal family, but there are various such examples which are associated with common people, and pedigree charts, pedigree analysis can be used to advise those people if their offspring are going to have any disorder. So coming to description of pedigree, what exactly pedigree means? Now, Mendel's methodical crosses were made possible by the ease with which mating between parental types could be controlled in peas. We already know that Mendel was able to perform controlled crosses and come to a certain conclusion that how genes are getting passed, which gene is dominant, which gene is recessive, and how it is getting passed from one generation to next generation. But in the case of humans, these crosses are not possible simply because of ethical reasons, and humans will not lend themselves to experimentation like these, as there cannot be a controlled mating in a civilized society. Secondly, the generation time of humans is very long as compared to that of peas, and so direct studies would not be feasible in case of humans for finding out the inheritance pattern of specific genes and alleles. Therefore, special methods are needed to be employed to analyze human inheritance patterns of specific genes and specific traits. The analysis of pedigree is basically a symbolic representation of family history, and it is a classical method of genetic analysis in humans. This representation of history goes back to many generations. More the data of previous generation, more ascertained we will be of the inheritance pattern of a particular trait of that particular family. The pedigree consists of a set of symbols which convey information about incidences of a trait in successive generations. So these symbols are very much important whenever a pedigree is being constructed, and a student of genetist must know about all these symbols in a proficient manner. These are the symbols which are used in constructing a pedigree chart. For denoting individuals of different sexes, that is for denoting male and female, we use different symbols. For male, a square is used, whereas for female, a circle is used. In case the sex of the individual is undesignated or unidentified, then a rhombus is used in that particular case. Individuals who are adopted, they would be represented by square which is enclosed in a parenthesis. Any pregnancy will be denoted by a rhombus, and a P letter is put inside that particular rhombus. Those individuals which die in a family history, they will be represented by square or circle with a diagonally crossing line present there. Individuals who are affected with that particular trait will be shaded. So if a male is affected or a female is affected, the square and circle will be completely shaded black. in case the individuals are heterozygous, that is they are carrier for a trait. In case of recessive traits, then half the part of square and half the part of circle will be shaded. Any carrier for X-linked trait, X is the chromosome of chromosome which is present in 2 copies in females and 1 in males. Any female who is a carrier for X-linked trait will be represented by a circle with a dark dot present in between. Mating between 2 individuals, between the male and female, will be represented by a connecting horizontal line between the circle and the square. Whenever there is consanguineous mating, that is if the cousins marry each other, that would be represented by 2 horizontal lines connecting the square and the circle. Siblings are represented by vertical lines emanating from a horizontal line, which is also known as ship line. So, all the siblings would be connected through vertical lines to a horizontal line, which is a ship line. The number of children can also be represented by putting in the numbers in squares or circles. Any couple which has divorced or separated would be represented by a cross in between the marriage line. Dizygotic twins, that is fraternal twins, would be represented by two lines which are emanating from a common point. And these dizygotic twins could be male, could be female, or both male and female. However, monozygotic twins, that is identical twins, they would be represented by 2 circles or 2 squares which are connected through a common point, but these 2 circles or 2 squares would be joined together by a horizontal line. In case the marriage does not result in production of any offspring, then it would be represented by number of horizontal lines under the vertical line emanating from the marriage line. Any individual who first comes into the focus of the physician, who first recognizes a particular trait, would be represented by an arrow and is termed as proposita or propositus. Propositus for male and proposita for female. If an individual has 2 matings, it would be represented, but the marriage which has been abandoned would be represented by a cross. So, these are the symbols which will be used for constructing a pedigree chart in any of the family in which we are studying inheritance of a given trait. While we are constructing a pedigree chart, there are certain points to be remembered, and these 2 important points are that every generation of a pedigree chart will be assigned a Roman numeral and will be starting from the top. So, the most ancient generation or the would be represented by Roman numeral 1, their offspring would be represented by 2, followed by their offspring which would be represented by 3, 4 and so on. Also, in each generation we would be assigning a number to all of the individuals starting from the left side. So, in this particular case we see that in generation 1, 4 individuals have been accounted for, and in first generation the leftmost is a male who is affected because he is completely because the square is completely shaded. That individual is numbered 1. The partner of that male is numbered 2, and right to that is a female who would be numbered 3, who is married to a male numbered 4. So, we would be numbering all the individuals in each generation for carrying out this particular pedigree analysis. This gives us ease of finding out which individual is having which particular genotype. Now, using carefully constructed pedigrees, one can determine whether a given phenotype is expressed because of a dominant or a recessive allele, and also it can be used to find out whether the trait is sex linked. There are 5 different types of inheritance patterns which can be ascertained from the pedigree chart, and these inheritance patterns are: whenever there is autosomal dominant inheritance, autosomal recessive inheritance, X-linked dominant inheritance, X-linked recessive inheritance, and Y-linked inheritance. I will be talking about all of these inheritance patterns one by one. So, the first one is autosomal dominant. That means genes are residing on autosomes, and the inheritance pattern, the trait which is being shown is because of presence of a dominant allele. Whenever a copy of dominant allele is present, this trait would be expressed. But if two recessive alleles are present, this particular trait would not be expressed. So, in autosomal dominant inheritance, every affected individual has an affected biological parent. Secondly, there is no skipping of generation. That means this particular trait will be seen in all the generations. Males and females would have an equally likely chance of being affected with this particular trait. There would not be any differentiation between males and females in this particular trait because it is not linked to any of the sex chromosome. Instead, it is linked to autosomes. The recurrence risk of each child of an affected parent is 1 by 2, that is 0.5 is the probability of recurrence risk of all the children. Now, normal siblings of an affected individual do not transmit the trait to their offspring. The siblings which are normal signify that they are recessive homozygous, they do not have a copy of dominant allele, and they will not be able to transmit the trait to their offspring. Now, this is one of an example of a pedigree which shows autosomal dominant inheritance. In this particular case, we see shaded individuals, that is black individuals which are affected, black squares and black circles which denote individuals who are affected, and unshaded circles and squares which denote individuals who are not affected. So, we have to assign genotypes to these individuals which are represented in shaded and unshaded circles and squares. How we assign that? All unaffected are two lowercase alleles, that is small d, they are recessive homozygous. So, the numbers of those individuals in the pedigree chart have been given. Unaffected individuals are recessive homozygous. Affected children of an affected parent and an unaffected parent must be heterozygous, as they inherited a small d allele from the unaffected parent. The affected parents of an unaffected child must be heterozygous, capital D, small d, since they both passed a small d allele to their child. If both parents are heterozygous, that is capital D, small d, then their affected offsprings have a 2 out of 3 chance of being capital D, small d, and a 1 out of 3 chance of being capital D and capital D. The outsider rule for dominant autosomal pedigrees is that an affected outsider, that is a person with no known parents who marries into the family, is assumed to be heterozygous, that is capital D, small d. And any outsider who marries into this particular family showing the trait, and this particular person is also showing the trait, this individual would be considered as a heterozygous individual denoting capital D and small d. So, these are the rules of assigning genotype to autosomal dominant individual. There are many traits or diseases or symptoms which result from autosomal dominant pattern of inheritance, and the example of this particular inheritance pattern is achondroplasia, brachydactyly, Huntington's disease, myotonic dystrophy, familial hypercholesterolemia. These are some of the diseases which are caused because of a dominant allele. Now, the second mode of inheritance is that of autosomal recessive inheritance. As the name suggests, this particular inheritance pattern is dependent on presence of recessive homozygous condition in the individual. Only those individuals who are having recessive alleles in double copy would be able to express the trait. All the other individuals, whether they are having double copy of dominant allele or one copy of dominant allele and one copy of recessive allele, they will not be expressing this particular trait. So, what are the characteristics of autosomal recessive inheritance? Males and females are equally likely to be affected in this particular mode of inheritance. If both the parents are affected, then all the children are affected, because if both parents are affected, it means both are recessive homozygous individuals. They can only form gametes which are having recessive allele, and when these recessive alleles, any ovum bearing recessive allele and any sperm bearing recessive allele, when they fuse together, they would give rise to zygote which are having double copy of recessive allele. So, all the children will be affected. On average, the recurrence risk to the unborn sibling of an affected individual in case of autosomal recessive inheritance is 1 out of 4, that is 0.25. This particular trait is characteristically found in siblings and not in the parents of affected or the offspring of the affected generation. So, this particular mode of inheritance of trait skips generation. There might be generation which do not show any, in which any of the individual do not show the trait, and suddenly in the next generation this trait might appear. The parents of affected children may be related. The rarer the trait in the general population, the more likely a consanguineous mating is involved. So, whenever we are talking of recessive autosomal disease, it is to be noted that it is found in greater probability in those families where marriages of cousins are allowed, because that particular family will be having a higher chance of carrying that particular recessive allele, and when the cousins marry, the offspring which are formed will be having recessive homozygous condition. So, usually it is not advised for the cousins to get married. Now, if we look into this particular pedigree chart of autosomal recessive inheritance, we see that there are again shaded and unshaded squares and circles. Each generation has been numbered, and we see a particular pattern in second generation. In the second generation, the 8 and 9 individuals, 8 is a female and 9 is a male, they do not show this particular condition, whereas in the third generation their offspring, namely 15, 16 do not show this particular condition, but the offspring 17 start showing this particular trait. This suggests that there is a skipping of generation, though the parents do not show that particular trait, but their offspring are showing, one of their offspring is showing that particular trait, which points towards inheritance pattern which is autosomal recessive. So, how do we assign genotypes for this type of pedigree? All the affected individuals are 2 lowercase alleles, that is recessive alleles in homozygous condition. If an affected person mates with an unaffected person, any unaffected offspring which is thus produced must be heterozygous, that is capital R and small r. If all the offsprings are unaffected, then unaffected parent can be either capital R or capital R small r. If the offsprings are affected as well as unaffected, as we saw in this particular pedigree, then the unaffected parents ought to be heterozygous. Only heterozygous parents can produce progeny which are either affected or unaffected. If two unaffected individuals mate and have an affected child, then both parents must be heterozygous. Children of capital R and capital R small r have 1/2 chance of being capital R and 1/2 chance of being capital R and small r. Unaffected children of capital R small r crossed with capital R small r have a 2/3 chance of being heterozygous, that is capital R small r, and 1 out of 3 chance of being capital R. The outsider rule for recessive autosomal inheritance is that unaffected outsiders are assumed to be capital R, that is homozygous normal. If any individual is marrying into a family which is showing this particular trait and that individual is not affected, we will assume that individual not to be heterozygous, but instead will be assuming that particular individual to be homozygous. There are many diseases which occur because of autosomal recessive pattern of inheritance, and the example are cystic fibrosis, phenylketonuria, sickle cell anemia, Tay-Sachs disease, Alzheimer's disease and albinism. Now I come to the third mode of inheritance, which is X-linked dominant inheritance. As the name suggests, this particular inheritance is linked to X chromosome, and the trait shows when the allele is dominant. Any individual having a dominant allele will be showing this particular trait. So every affected individual in this particular mode of inheritance has an affected biological parent. There is no skipping of generations. Males always inherit trait from their mother, because males always get X from their mother and Y from the father. So males will be inheriting this particular trait from their mother. Same inheritance pattern as autosomal dominant trait is seen in human female. All the daughters of a male who has this particular trait will also have the trait, because all the daughters get one X from their father, and the daughters, even if she is getting a normal allele, X with a normal allele from the mother, if she gets any X chromosome from the father which is having a affected allele, dominant allele, then she will be showing this particular trait. So all the daughters of affected male will be showing this particular trait. There is no male to male transmission, because male only contribute Y chromosome to their son, and the trait follows the inheritance pattern of the X chromosome. This is the particular pedigree which can be ascertained to be that of X-linked dominant inheritance. Here again we can ascertain that it is X-linked dominant inheritance because we are able to assign genotypes to individuals. All the affected males are XAY. All unaffected females will have genotype XA, that is small xa. All unaffected males will be having X small a and Y. If an affected female mate with an unaffected male and all the offsprings are affected, then it means that the affected female parent was X capital A, that means both the alleles were dominant. If the offspring are affected and unaffected males and females, then the affected female parent is heterozygous individual, that is X capital A and X small a. The affected female progeny would also be heterozygous, that is X capital A and X small a. If an unaffected female X small a mate with an affected male, that is X capital A and Y, all the unaffected male progeny will be X small a and Y, and all the affected female progeny will be heterozygous, that is X capital A and X small a. Examples of X-linked dominant diseases are like congenital generalized hypertrichosis, Werewolf syndrome, which is signified by a large number of hair in the body. The next pattern of inheritance is X-linked recessive inheritance, which again is because of recessive homozygous condition of alleles and which are present on X chromosome. This trait is more common in males than in females. Males always inherit trait from their mother. If a female is affected, all her male offspring would be affected. Same inheritance pattern as autosomal recessive trait in human females can be seen in this particular pattern. There is no male to male transmission of X-linked recessive traits, and the trait can skip generation. This particular pedigree chart is that of X-linked recessive inheritance. How do we assign genotype to X-linked recessive inheritance? All affected males are X small a Y, all unaffected males are X capital A Y. All affected females will be X small a. If an affected female mates with an affected male, all the offspring will be affected. If an affected female mates with an unaffected male, then the female progeny will be heterozygous, that is a carrier X capital A and X small a. If a carrier female mates with an unaffected male, the unaffected female offspring have a 1 out of 2 chance of being a carrier, XA, X small a, and a 1 out of 2 chance of being X capital A. 1/2 of the male progeny will be affected in this particular inheritance. The examples of X-linked recessive inheritance are like hemophilia, muscular dystrophy and fragile X syndrome, which are very commonly seen in families families which are having this particular trait. Next type of inheritance is Y-linked inheritance, where trait expression and transmission is only in males, the individuals with the Y chromosome. Females do not express these traits. If a male has a trait, so should his father and paternal grandfather, as well as his sons and their sons. It follows the inheritance pattern of Y chromosome. This is an example of Y-linked pedigree. This particular pedigree chart shows that only the males are colored or shaded. So, because Y chromosome is inherited from father to son, we assign genotypes to in this particular manner. All affected males are XYA, all females are unaffected XX, and all unaffected males are XY small a. There are some traits like hairy ears and retinitis pigmentosa which follow this particular pattern of inheritance. Now, this pedigree analysis is a very useful tool in genetics and medical field, because it can be used by medical practitioners to assess the probability of an individual inheriting a specific trait in a given family. Now, I'll be we'll be doing one pedigree analysis problem. This is a particular pedigree, and we have to find out what is the mode of inheritance in this particular case. We see that there are individuals who are affected. We see in second generation the rightmost individual, which is a female, is affected, whereas the parents of this particular individual are not affected, they are not shaded. So, this individual must be small a. Because the parents are not affected, that means they must be heterozygous individuals. So, both will be capital A and small a, whereas all the other individuals who are affected are small a. The parents of affected individuals are capital A, small a. These siblings of this particular individual, since they do not show the trait and the parents are heterozygous, these individuals must also be either heterozygous or homozygous for the dominant allele. But this particular individual who is married to an affected individual, when he marries this particular female, it leads to formation of two sons who are affected, which means that this particular individual cannot be capital A, which means this individual is a heterozygous individual, capital A and small a. So, all in all, this points towards this particular pedigree being a autosomal recessive pedigree. Likewise, we can solve many pedigree problems by assigning genotypes to different individuals of different generation, and this will help us, the students of genetics, a great deal. Thank you.
On that note, I would like to thank Sir for this very enriching discussion, and thank you dear friends for watching our show. Stay tuned and keep watching. Thank you.